Plague is an infectious disease caused by the bacterium Yersinia pestis. 2. Symptoms include fever, weakness and headache. 1. Usually this begins 1 to 7 days after exposure. 2. There are 3 forms of plague, each affecting a different part of the body and causing associated symptoms. Pneumonic plague infects the lungs, causing shortness of breath, coughing and chest pain. Bubonic plague affects the lymph nodes, making them swell, and septicemic plague infects the blood and can cause tissues to turn black and die. 2. 1. The bubonic and septicemic forms are generally spread by flea bites or handling an infected animal. 1. Whereas pneumonic plague is generally spread between people through the air via infectious droplets. 1. Diagnosis is typically by finding the bacterium in fluid from a lymph node, blood or sputum. 2. Those at high risk may be vaccinated. 2. Those exposed to a case of pneumonic plague may be treated with preventive medication. 2. If infected, treatment is with antibiotics and supportive care. 2. Typically antibiotics include a combination of gentamicin and a fluoroquinolone. 3. The risk of death with treatment is about 10% while without it is about 70%. 4. Globally, about 600 cases are reported a year. 2. In 2017, the countries with the most cases include the Democratic Republic of the Congo, Madagascar and Peru. 2. In the United States, infections occasionally occur in rural areas, where the bacteria are believed to circulate among rodents. 5. It has historically occurred in large outbreaks, with the best known being the Black Death in the 14th century, which resulted in more than 50 million deaths in Europe. 2. Signs and symptoms. There are several different clinical manifestations of plague. The most common form is bubonic plague, followed by septicemic and pneumonic plague. 6. Other clinical manifestations include plague meningitis, plague pharyngitis, and ocular plague. 6. 7. General symptoms of plague include fever, chills, headaches, and nausea. 1. Many people experience swelling in their lymph nodes if they have bubonic plague. 1. For those with pneumonic plague, symptoms may or may not include a cough, pain in the chest, and hemoptysis. 1. Bubonic plague. Swollen inguinal lymph glands on a person infected with the bubonic plague. The swollen lymph glands are termed bubos from the Greek word for groin, swollen gland, bubo. When a flea bites a human and contaminates the wound with regurgitated blood, the plague-causing bacteria are passed into the tissue. Y. Pestis can reproduce inside cells, so even if phagocytosed, they can still survive. Once in the body, the bacteria can enter the lymphatic system, which drains interstitial fluid. Plague bacteria secrete several toxins, one of which is known to cause beta-adrenergic blockade. 8. Y. pestis spreads through the lymphatic vessels of the infected human until it reaches a lymph node, where it causes acute lymphadenitis. 9. The swollen lymph nodes form the characteristic buboes associated with the disease. 10. An autopsies of these buboes have revealed them to be mostly hemorrhagic or necrotic. 11. If the lymph node is overwhelmed, the infection can pass into the bloodstream, causing secondary septicemic plague, and if the lungs are seeded, it can cause secondary pneumonic plague. 12. Plague. Yersinia pestis seen at 200 times magnification. With a fluorescent label. Specialty infectious disease. Symptoms: fever, weakness. Headache. 1. Usual onset 1 to 7 days after exposure. 2. Types: bubonic plague, septicemic plague. Pneumonic plague. 1. Causes: Yersinia pestis. 2. Diagnostic method: finding the bacterium in a lymph node, blood, sputum. 2. Prevention: plague vaccine. 2. Treatment: antibiotics and supportive care. 2. Medication: gentamicin and a fluoroquinolone. 3. Prognosis: approximately equals 10% risk of death with treatment. 4. Septicemic plague. Septicemic plague resulting in necrosis. Lymphatics ultimately drain into the bloodstream, so the plague bacteria may enter the blood and travel to almost any part of the body. In septicemic plague, bacterial endotoxins cause disseminated intravascular coagulation, DIC, causing tiny clots throughout the body and possibly ischemic necrosis, tissue death due to lack of circulation, perfusion to that tissue from the clots. DIC results in depletion of the body's clotting resources so that it can no longer control bleeding. Consequently, there is bleeding into the skin and other organs, which can cause red and/or black patchy rash and hemoptysis, hematemesis, coughing up, vomiting of blood. There are bumps on the skin that look somewhat like insect bites. These are usually red, and sometimes white in the center. Untreated, the septicemic plague is usually fatal. Early treatment with antibiotics reduces the mortality rate to between 4 and 15%. 13, 14, 15. Pneumonic plague. The pneumonic form of plague arises from infection of the lungs. It causes coughing and thereby produces airborne droplets that contain bacterial cells and are likely to infect anyone inhaling them. The incubation period for pneumonic plague is short, usually 2 to 4 days, but sometimes just a few hours. The initial signs are indistinguishable from several other respiratory illnesses. They include headache, weakness, and spitting or vomiting of blood. The course of the disease is rapid. Unless diagnosed and treated soon enough, typically within a few hours, death may follow in 1 to 6 days. In untreated cases, mortality is nearly 100%. 16, 17. Frequency approximately equals 600 cases a year. 2. Cause. The oriental rat flea, Xenopsila cheapus, engorged with blood after a blood meal. This species of flea is the primary vector for the transmission of Yersinia pestis, the organism responsible for bubonic plague in most plague epidemics in Asia, Africa, and South America. Both male and female fleas feed on blood and can transmit the infection. A child bitten by a flea infected with the bacterium Yersinia pestis. Y. pestis, a member of the family Yersineaceae, has caused the bite to become ulcerated. Transmission of Y. pestis to an uninfected individual is possible by any of the following means. 18. Droplet contact, coughing or sneezing on another person. Direct physical contact, touching an infected person, including sexual contact and direct contact, usually by touching soil contamination or a contaminated surface. Airborne transmission, if the microorganism can remain in the air for long periods. Fecal oral transmission, usually from contaminated food or water sources. Vector-borne transmission, carried by insects or other animals. Yersinia pestis circulates in animal reservoirs, particularly in rodents, in the natural foci of infection found on all continents except Australia. The natural foci of plague are situated in a broad belt in the tropical and subtropical latitudes and the warmer parts of the temperate latitudes around the globe, between the parallels 55 degrees north and 40 degrees S. 1-8. Contrary to popular belief, rats did not directly start the spread of the bubonic plague. It is mainly a disease in the fleas, Xenopsila cheapus, that infested the rats, making the rats themselves the first victims of the plague. Rodent-borne infection in a human occurs when a person is bitten by a flea that has been infected by biting a rodent that itself has been infected by the bite of a flea carrying the disease. The bacteria multiply inside the flea, sticking together to form a plug that blocks its stomach and causes it to starve. The flea then bites a host and continues to feed, even though it cannot quell its hunger, and consequently, the flea vomits blood tainted with the bacteria back into the bite wound. The bubonic plague bacterium then infects a new person and the flea eventually dies from starvation. Serious outbreaks of plague are usually started by other disease outbreaks in rodents or a rise in the rodent population. 19. A 21st century study of a 1665 outbreak of plague in the village of Ayham in England's Derbyshire Dales, which isolated itself during the outbreak, facilitating modern study, found that 3/4 of cases are likely to have been due to human transmission, especially within families, a much larger proportion than previously thought. 20. Diagnosis. Symptoms of plague are usually nonspecific, and to definitively diagnose plague, laboratory testing is required. 21. Y. pestis can be identified through both a microscope and by culturing a sample, and this is used as a reference standard to confirm that a person has a case of plague. 21. The sample can be obtained from the blood, mucus, sputum, or aspirate extracted from inflamed lymph nodes, buboes. 21. If a person is administered antibiotics before a sample is taken, or if there is a delay in transporting the person's sample to a laboratory and/or a poorly stored sample, there is a possibility for false negative results. 21. Polymerase chain reaction (PCR) may also be used to diagnose plague by detecting the presence of bacterial genes such as the PLA gene, plasmogene activator, and CAF1 gene, F1 capsule antigen. 21. PCR testing requires a very small sample and is effective for both alive and dead bacteria. 21. For this reason, if a person receives antibiotics before a sample is collected for laboratory testing, they may have a false negative culture and a positive PCR result. 21. Blood tests to detect antibodies against Y. pestis can also be used to diagnose plague. However, this requires taking blood samples at different periods to detect differences between the acute and convalescent phases of F1 antibody titers. 21. In 2020, a study about rapid diagnostic tests that detect the F1 capsule antigen, F1-RDT, by sampling sputum or bubo aspirate was released. 21. Results show rapid diagnostic F1-RDT test can be used for people who have suspected pneumonic and bubonic plague but cannot be used in asymptomatic people. F1-RDT may be useful in providing a fast result for prompt treatment and fast public health responses. Studies suggest that F1-RDT is highly sensitive for both pneumonic and bubonic plague. However, when using the rapid test, both positive and negative results need to be confirmed to establish or reject the diagnosis of a confirmed case of plague, and the test result needs to be interpreted within the epidemiological context, as study findings indicate that although 40 out of 40 people who had the plague in a population of 1,000 were correctly diagnosed, 317 people were diagnosed falsely as positive. 21, 22, 23. Prevention. Vaccination. Bacteriologist Waldemar Haffkine developed the first plague vaccine in 1897. 24-25. He conducted a massive inoculation program in British India, and it is estimated that 26 million doses of Haffkine's anti-plague vaccine were sent out from Bombay between 1897 and 1925, reducing the plague mortality by 50-85%. 24-26. Since human plague is rare in most parts of the world as of 2023, routine vaccination is not needed other than for those at particularly high risk of exposure, nor for people living in areas with enzootic plague, meaning it occurs at regular, predictable rates in populations in specific areas, such as the western United States. It is not even indicated for most travelers to countries with known recent reported cases, particularly if their travel is limited to urban areas with modern hotels. The United States CDC thus only recommends vaccination for 1. All laboratory and field personnel who are working with Y. pestis organisms resistant to antimicrobials, 2. People engaged in aerosol experiments with Y. pestis, and 3. People engaged in field operations in areas with enzootic plague where preventing exposure is not possible, such as some disaster areas. 27. A systematic review by the Cochrane Collaboration found no studies of sufficient quality to make any statement on the efficacy of the vaccine. 28. Early diagnosis. Diagnosing plague early leads to a decrease in transmission or spread of the disease. 21. Prophylaxis. Pre-exposure prophylaxis for first responders and health care providers who will care for patients with pneumonic plague is not considered necessary as long as standard and droplet precautions can be maintained. 7. In cases of surgical mask shortages, patient overcrowding, poor ventilation in hospital wards, or other crises, pre-exposure prophylaxis might be warranted if sufficient supplies of antimicrobials are available. 7. Post-exposure prophylaxis should be considered for people who had close, less than 6 feet, sustained contact with a patient with pneumonic plague and were not wearing adequate personal protective equipment. 7. Antimicrobial post-exposure prophylaxis also can be considered for laboratory workers accidentally exposed to infectious materials and people who had close, less than 6 feet, or direct contact with infected animals, such as veterinary staff, pet owners, and hunters. 7. Specific recommendations on pre- and post-exposure prophylaxis are available in the Clinical Guidelines on Treatment and Prophylaxis of Plague published in 2021. 7. Treatments. If diagnosed in time, the various forms of plague are usually highly responsive to antibiotic therapy. 621. The antibiotics often used are streptomycin, chloramphenicol and tetracycline. Amongst the newer generation of antibiotics, gentamicin and doxycycline have proven effective in monotherapeutic treatment of plague. 629. Guidelines on treatment and prophylaxis of plague were published by the Centers for Disease Control and Prevention in 2021. 7. The plague bacterium could develop drug resistance and again become a major health threat. One case of a drug-resistant form of the bacterium was found in Madagascar in 1995. 30. Further outbreaks in Madagascar were reported in November 2014, 31, and October 2017. 32. Epidemiology. Distribution of plague-infected animals 1998. Globally about 600 cases are reported a year. 2. In 2017, the countries with the most cases include the Democratic Republic of the Congo, Madagascar and Peru. 2. It has historically occurred in large outbreaks, with the best known being the Black Death in the 14th century which resulted in more than 50 million dead. 2. In recent years, cases have been distributed between small seasonal outbreaks which occur primarily in Madagascar, and sporadic outbreaks or isolated cases in endemic areas. 2. In 2022 the possible origin of all modern strands of Yersinia pestis DNA was found in human remains in 3 graves located in Kyrgyzstan, dated to 1338 and 1339. The siege of Kaffa in Crimea in 1346 is known to have been the first plague outbreak with following strands, later to spread over Europe. Sequencing DNA compared to other ancient and modern strands paints a family tree of the bacteria. Bacteria today affecting marmots in Kyrgyzstan are closest to the strand found in the graves, suggesting this is also the location where plague transferred from animals to humans. 33. Biological weapon. The plague has a long history as a biological weapon. Historical accounts from ancient China and medieval Europe details the use of infected animal carcasses, such as cows or horses, and human carcasses, by the Xiongnu, Huns, Mongols, Turks and other groups, to contaminate enemy water supplies. Han Dynasty General Huo Qubing is recorded to have died of such contamination while engaging in warfare against the Xiongnu. Plague victims were also reported to have been tossed by catapult into cities under siege. 34. In 1347, the Genoese possession of Kaffa, a great trade emporium on the Crimean Peninsula, came under siege by an army of Mongol warriors of the Golden Horde under the command of Yani Beg. After a protracted siege during which the Mongol army was reportedly withering from the disease, they decided to use the infected corpses as a biological weapon. The corpses were catapulted over the city walls, infecting the inhabitants. This event might have led to the transfer of the Black Death via their ships into the south of Europe, possibly explaining its rapid spread. The plague has a long history as a biological weapon. Historical accounts from ancient China and medieval Europe details the use of infected animal carcasses, such as cows or horses, and human carcasses, by the Xiongnu, Huns, Mongols, Turks and other groups, to contaminate enemy water supplies. Han Dynasty General Huo Qubing is recorded to have died of such contamination while engaging in warfare against the Xiongnu. Plague victims were also reported to have been tossed by catapult into cities under siege. 34. In 1347, the Genoese possession of Kaffa, a great trade emporium on the Crimean Peninsula, came under siege by an army of Mongol warriors of the Golden Horde under the command of Yani Beg. After a protracted siege during which the Mongol army was reportedly withering from the disease, they decided to use the infected corpses as a biological weapon. The corpses were catapulted over the city walls, infecting the inhabitants. This event might have led to the transfer of the Black Death via their ships into the south of Europe, possibly explaining its rapid spread. 35. During World War II, the Japanese army developed weaponized plague, based on the breeding and release of large numbers of fleas. During the Japanese occupation of Manchuria, Unit 731 deliberately infected Chinese, Korean and Manchurian civilians and prisoners of war with the plague bacterium. These subjects, termed maruta, or logs, were then studied by dissection, others by vivisection while still conscious. Members of the unit such as Shiro Ishii were exonerated from the Tokyo Tribunal by Douglas MacArthur, but 12 of them were prosecuted in the Khabarovsk war crime trials in 1949, during which some admitted having spread bubonic plague within a 36-kilometer (22 miles) radius around the city of Changde. 36. Ishii innovated bombs containing live mice and fleas, with very small explosive loads, to deliver the weaponized microbes, overcoming the problem of the explosive killing the infected animal and insect by the use of a ceramic, rather than metal, casing for the warhead. While no records survive of the actual usage of the ceramic shells, prototypes exist and are believed to have been used in experiments during World War II. 37-38. After World War II, both the United States and the Soviet Union developed means of weaponizing pneumonic plague. Experiments included various delivery methods, vacuum drying, sizing the bacterium, developing strains resistant to antibiotics, combining the bacterium with other diseases, such as diphtheria, and genetic engineering. Scientists who worked in USSR bioweapons programs have stated that the Soviet effort was formidable and that large stocks of weaponized plague bacteria were produced. Information on many of the Soviet and US projects is largely unavailable. Aerosolized pneumonic plague remains the most significant threat. 39-40-41, the plague can be easily treated with antibiotics. Some countries, such as the United States, have large supplies on hand if such an attack should occur, making the threat less severe. 42. See also. Timeline of plague. References. 1. Strepsiptera. The Strepsiptera, starpster, are an order of insects with 11 extant families that include about 600 described species. They are endoparasites of other insects, such as bees, wasps, leafhoppers, silverfish, and cockroaches. Females of most species never emerge from the host after entering its body, finally dying inside it. The early stage larvae do emerge because they must find an unoccupied living host, and the short-lived males must emerge to seek a receptive female in her host. 1. They are believed to be most closely related to beetles, from which they diverged 300 to 350 million years ago, but do not appear in the fossil record until the mid-Cretaceous around 100 million years ago. 2. The order is not well known to non-specialists, and the nearest they have to a common name is Stylops, in reference to the genus Stylops. 3. The name of the order translates to twisted wing, giving rise to other common names used for the order, twisted wing insects and twisted wing parasites. 4. Adult males are rarely observed, although specimens may be lured using cages containing virgin females. Nocturnal specimens can also be collected at light traps. 1. Biology. Appearance and structure. Males. Males of the Strepsiptera have wings, legs, eyes, and antennae, though their mouthparts cannot be used for feeding. Many have mouthparts modified into sensory structures. The males bear a superficial resemblance to flies. 1. The forewings are modified into small club-shaped structures called halteres, which sense gyroscopic information. 5. A similar organ exists in flies, though in that group the hindwings are modified instead, and the two groups are thought to have independently evolved the structures. 6. The hindwings are generally fan-shaped, and have strongly reduced venation. The antennae are flavolate, and are covered in specialized chemoreceptors, likely to detect females over long distances. 7. Adult male Strepsiptera have eyes unlike those of any other insect, resembling the eyes found in the trilobite group Phacopina. Instead of a compound eye consisting of hundreds to thousands of omat. 7. Adult male Strepsiptera have eyes unlike those of any other insect, resembling the eyes found in the trilobite group Phacopina. Instead of a compound eye consisting of hundreds to thousands of ommatidia, that each produce a pixel of the entire image, the Strepsipteran eyes consist of only a few dozen islets, that each produce a complete image. These islets are separated by cuticle and or setae, giving the cluster eye as a whole a blackberry-like appearance. 1-8. Females. The females of Stylopidia, which includes 97% of all described Strepsipteran species and all modern Strepsipteran families except Mengenilidae and Bahyaxinidae, are not known to leave their hosts and are neotenic in form, lacking wings, legs, and eyes, but have a well-sclerotized cephalothorax, fused head and thorax. 1-9-7. Adult female Mengenilids are wingless but are free-living and somewhat mobile with legs and small eyes. This is probably also true for the Bahyaxinids, though this has not been observed. 10-9-7. Larva. Newly hatched primary first instar larvae are on average 230 micrometers, 1/128th in length, smaller than many single-celled organisms. They are highly mobile with well-developed stemata, which are able to distinguish color. The underside of the body is covered in minute hair-like structures, microtrichia, which allow the larva to stick to wet surfaces via capillary action. At the back of the body are well-developed large bristle-like cerci, which are attached to muscles, which allow the larva to jump. The tarsal segment of their legs have structures which allow them to cling to their hosts. Later larval instars which develop inside the host are completely immobile. 7. Strepsiptera. Temporal range, Middle Cretaceous recent. Male. Stylopid female. Scientific classification domain, Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta unranked, Holometabola clade, Aparaglossata clade, Neuropteroidea. Free-living female of Mengenilla moldricki, Mengenilidae. Male pupa head top left and adult female of Xenos yangi, Stylopidia, Xenidae in ventral view, right, and close-up of the cephalothorax, center and bottom left. Clade, Coleopterida order, Strepsiptera. Curve. Recently hatched larva of a stylopid Strepsipteran on the B. Andrena nivalis. Close-up of the head of male Xenos pecci. Stylops melite, Stylopidae, female Stylopizing B. Andrena vega, exposing its cephalothorax. Adult male Strepsipteran in dorsal view, halteres highlighted with red arrows. Life cycle. Traumatic insemination of an endoparasitic female in Stylops ovinae. Virgin females release a pheromone which the males use to locate them. 1. Traumatic insemination of an endoparasitic female in Stylops ovinae. Virgin females release a pheromone which the males use to locate them. 1. Mating in at least some species is polyandrous, where the female mates with more than one male. 11. In the Stylopidia, the female's anterior region protrudes out between the segments of the host's abdomen. In all strepsipterans, the male mates by rupturing the female's cuticle. In the case of Stylopidia, this is in a deep narrow fissure of the cephalothorax near the birth canal. Sperm passes through the opening directly into the body in a process called traumatic insemination, which has independently evolved in some other insects like bedbugs. 1.11. Strepsiptera eggs hatch inside the female, and the planidium larva can move around freely within the female's hemocoel. This behavior is unique to these insects. 12. The offspring consume their mother from the inside in a process known as hemocoelous viviparity. Each female produces many thousands of planidium larvae. 13. The larvae emerge from the brood opening, canal on the female's head, which protrudes outside the host body. 12.13. Larvae have legs and actively seek out new hosts. Their legs are partly vestigial in that they lack a trochanter, the leg segment that forms the articulation between the basal coxa and the femur. 13. The larvae are very active as they only have a limited amount of time to find a host before they exhaust their energy reserves. These first instar larvae have stemata, simple, single lens eyes. When the larvae latch onto a host, they enter it by secreting enzymes that soften the cuticle, usually in the abdominal region of the host. Some species have been reported to enter the eggs of hosts. Larvae of Stichotrema dalatornernhoffender from Papua New Guinea were found to enter their orthopteran host's tarsus foot. 14. Once inside the host, they undergo hypermetamorphosis and transform into a less mobile, legless larval form. They induce the host to produce a bag-like structure inside which they feed and grow. This structure, made from host tissue, protects them from the immune defenses of the host. Larvae go through 4 more instars, and in each molt the older cuticle separates but is not discarded, a polycis without ecdysis, so multiple layers form around the larva. 15. Male larvae pupate after the last molt, but females directly become neotenous adults. 16. 17. The color and shape of the host's abdomen may be changed and the host usually becomes sterile. The parasites then undergo pupation to become adults. Adult males emerge from the host bodies, while females stay inside. Females may occupy up to 90% of the abdominal volume of their hosts. 1. Adult males are very short-lived, usually surviving less than 5 hours, and do not feed. 1. Parasitism. Strepsiptera of various species have been documented to attack hosts in many orders, including members of the orders Zygentoma, silverfish and allies, Orthoptera, grasshoppers, crickets, Blatodia, cockroaches. Strepsiptera of various species have been documented to attack hosts in many orders, including members of the orders Zygentoma, silverfish and allies, Orthoptera, grasshoppers, crickets, Blatodia, cockroaches, Mantodia, praying mantis, Heteroptera, bugs, Hymenoptera, wasps, ants and bees, and Diptera, flies. In the Strepsipteran family Myrmeculacidae, the males parasitize ants, while the females parasitize Orthoptera. 1. Members of Mengenilidae target Zygentoma exclusively. 18. While Stylopidia targets only winged insects, with a large number of Stylopodians targeting wasps and bees, while the largest family of Strepsipterans, the Stylopidae, with over 27% of all described Strepsipterans, targets bees exclusively. 7. Very rarely, multiple females may live within a single stylopized host. Multiple males within a single host are somewhat more common. 1. Strepsiptera of the family Myrmeculacidae can influence their host's behavior, causing their ant hosts to linger on the tips of grass leaves, increasing the chance of being found by Strepsipteran males, in the case of females, and putting them in a good position for male emergence, in the case of males. 19. Taxonomy. Mengenilla moldricki, Mengenilidae. The order, named by William Kirby in 1813, 20, is named for the hindwings, which are held at a twisted angle when at rest, from Greek straphion, strepine, to twist, and pteron, wing. 21. The forewings are reduced to halteres. A wasp, Odinurus sphenipes, with a small portion of a Strepsipteran's body protruding from its abdomen. Strepsiptera were once believed to be the sister group to the beetle families Meloidae and Rhipiphoridae, which have similar parasitic development and forewing reduction. Early molecular research suggested their inclusion as a sister group to the flies, 1. In a clade called Halteria, 22, which have one pair of the wings modified into halteres, 23, and failed to support their relationship to the beetles. 23. Further molecular studies, however, suggested they are outside the clade Mecopterida, containing the Diptera and Lepidoptera, but found no strong evidence for affinity with any other extant group. 24. Study of their evolutionary position has been problematic due to difficulties in phylogenetic analysis arising from long branch attraction. 25. Most modern molecular studies find Strepsipterans as the sister group of beetles Coleoptera, with both groups together forming the clade Coleopterida. 26. The most basal Strepsipteran is the fossil Protoxenos jansseni discovered in Eocene-aged Baltic amber. 27. While the most basal living Strepsipteran is Bahiochenos relictus, the sole member of the family Bahioxenidae. 28. The earliest known strepsipteran fossils are those of Cretostylops angeli, Cretostyloptii, and Kinzelbachilla ellenbergeri, Thynoxinose nervosus and Heterobathmillicocopoios, Thynoxinidae, discovered in Middle Cretaceous Burmese amber from Myanmar, around 99 million years old, which all lie outside the crown group, but are all more closely related to modern strepsiperans than Protoxinose is. The finding of a parasitic first instar in the same deposit indicates that the parasitic lifestyle of the group has likely existed nearly unchanged for 100 million years, though their evolutionary history prior to this remains a mystery. 2. The idea that Mengelinids' targeting of zygentomans represents the ancestral ecology of the group as a whole has been considered questionable. 18. Stylops melitae male. Stem group Strepsipteran Heterobathmillicocopoios. Thynoxinidae, in Burmese amber, around 100 million years old. Families. Andrena vega male bee, with Stylops melitae mating on its abdomen. The vast majority of living strepsisterans are placed within the grouping Stylopidae, which includes the families Corioxinidae, Halictophagidae, Callipharaxinidae, Bohartilidae, Elenchidae, Myrmeculacidae, Stylopidae, Protolencholucidae, extinct, and Xenidae. 2. All Stylopidae have endoparasitic females having multiple genital openings. 2. All Stylopidae have endoparasitic females having multiple genital openings. 1. 2 living families, Mengenilidae and Bahyaxinidae, are placed outside of this group, along with several extinct families. 2. The Stylopidae have 4 segmented tarsi and 4 to 6 segmented antennae, with the third segment having a lateral process. The family Stylopidae may be paraphyletic. 1. The Elenchidae have 2 segmented tarsi and 4 segmented antennae, with the third segment having a lateral process. The Halictophagidae have 3 segmented tarsi and 7 segmented antennae, with lateral processes from the third and fourth segments. 13. The Stylopidae mostly parasitize wasps and bees. The Elenchidae are known to parasitize Fulguroidea, while the Halictophagidae are found on leafhoppers, treehoppers, and mole cricket hosts. 13. Strepsipteran insects in the genus Xenos parasitize Polistes carnifex, a species of social wasps. 29. These obligate parasites infect the developing wasp larvae in the nest and are present within the abdomens of female wasps when they hatch out. Here they remain until they thrust through the cuticle and pupate, males, or release infective first instar larvae onto flowers, females. These larvae are transported back to their nests by foraging wasps. Protolencholucidae. Xenidae. Some insects which have been considered pests may have strepsipteran endoparasites. Inoculation of a pest population with the corresponding parasitoid may sometimes aid in reducing the impact of such pests, although no strepsipterans have ever been tested for use in this capacity, let alone being available for such purposes, either commercially or experimentally. Bizarre, and weird, are overused adjectives for describing many characters and events of the natural world. Life is way too complex and varied to conform to familiar patterns, so the out of the ordinary is all around us, even though we not always see it. But when the discussion turns to stylopids, it's difficult to avoid talking about the bizarre and the weird. Stylopids, or stylops, are small, seldom seen and poorly known insects with about 700 described species, 10 to 16 of them in Britain. The true figures are likely to be much higher. They are parasites of other insects such as bees, wasps, plant hoppers and leaf hoppers. From a distance, male stylopids can be mistaken for flies, but their ruffled wings give them away and explain the name of this group of insects. The order Strepsiptera, from the Greek strepine, to twist, and pteron, wing. Twisted wing insects is another common name for them. Males have branched antennae, and their eyes are berry-like structures comprising dozens of image-forming eyelets. This unusual array inspired the development of new cameras of reduced size and sharp images, which are handy for smartphones. Males cannot feed because their mouthparts are not developed. But never mind going hungry, they don't live for more than a few hours. Their only objective in life is to use their fancy eyes to find a female and mate. Females look nothing like the males. In fact, they don't look like your ordinary insect at all because they don't have wings, antennae, legs, mouthparts or eyes. They are neotenic, i.e., they retain their larval features. An adult female does not need a fully formed body since she never leaves her host. She will develop and die semi-buried in another insect. Semi-buried, because the tip of her cephalothorax, the head and the thorax fuse together, protrudes from the host's abdomen. Through this exposed area, the female releases a pheromone to attract males. Once suitors finds her, they face an anatomical challenge. Only parts of her head, thorax are exposed, which doesn't bode well for females look nothing like the males. In fact, they don't look like your ordinary insect at all because they don't have wings, antennae, legs, mouthparts or eyes. They are neotenic, i.e., they retain their larval features. An adult female does not need a fully formed body since she never leaves her host. She will develop and die semi-buried in another insect. Semi-buried, because the tip of her cephalothorax, the head and the thorax fuse together, protrudes from the host's abdomen. Through this exposed area, the female releases a pheromone to attract males. Once suitors finds her, they face an anatomical challenge. Only parts of her head, thorax are exposed, which doesn't bode well for conventional insect romance. But this setback is nothing compared to the facts that she doesn't have genitalia, and her eggs float in the hemolymph blood. So a male has only one course of action, the disturbingly sounding traumatic or hypodermic insemination. He pierces the female's cuticle with his penis and injects his sperm into her hemolymph. A Male Stylopod. Copyright Mike Quinn, Texacento.net, McKenna and Farrell, 2010, PLOS 157E11887, and detail of a male head copy. A B scale bars equal. The deed done, males soon die. The fertilized eggs hatch inside the female, giving birth to thousands of tiny six-legged, very active and agile larvae called planidia, just like blister or oil beetles. The planidia feed on mom's innards and eventually crawl out of her body to disperse and start looking for a host of their own. The planidia feed on mom's innards and eventually crawl out of her body to disperse and start looking for a host of their own. A wandering planidium climbs a flower to wait for an unsuspecting visitor. When a bee or wasp lands, the planidium somehow hitchhikes a ride to their nest. We are not sure how it does this. It could hide in the pollen, or possibly be swallowed with the nectar sucked up by the flower visitor, then released when the host regurgitates nectar inside the nest. Chances are it will end up in the wrong nest, so most planidia are done for. But the staggering fecundity of female stylopods compensates bad odds. They can dish out 750,000 planidia, so few are likely to find a suitable host. Once inside the right nest, the planidium burrows into a host's egg or larva and transforms into a traditional legless, grub-like larva. It is followed by other larval stages, pupation, and finally adulthood. By then the host has also become an adult. If it's a male stylopod, it squeezes out of the host and flies away, usually leaving a big gap behind and killing the host. If it's a female, it will park itself in the host's abdomen. A stylopod planidium, and planidia emerging from a female stylopod. Scale bars equals 0.1 mm. Copyright Cathirathambi, J. 2018. Biodiversity of Strepsiptera. A male stylopod emerging from a wasp. Copyright Cathirathambi, J. 2009. Annual Review of Entomology 54, 227-49. The story above is a peep at stylopods' life histories, as there is considerable variation depending on the species and type of host. And if all this sounds outlandish, there is more to come. Stylopization. Parasitism by stylopods causes all sorts of physical and behavioral eccentricities in the host, all for the parasite's benefit. Such as infertility. Reproduction involves mating, nest building, nest provision, etc., which are risky and energy-consuming, therefore not beneficial for a parasite. Stylopods solve this problem by disabling the host's reproductive organs, functionally castrating them. Some stylopized bees have reduced scopi pollen-carrying structures, and seldom if ever carry pollen. There's no point, as they don't have a brood to provide for. Contrary to what happens to most parasitized insects, stylopization often lengthens hosts' development. They live longer so that stylopods have more time to mature. Some stylopized bees are led to stand still on a grass or flower stems with their head downwards. Such a zombie state greatly facilitates stylopods' mating business. Some stylopized bees are led to stand still on a grass or flower stems with their head downwards. Such a zombie state greatly facilitates stylopods' mating business. In Britain, furrow bees, genera Halictus and Lasioglossum, yellow face bees, genus Hylias, and especially mining bees, genus Andrena, are victims of stylopods, but we have little information about their interactions and no idea about consequences of parasitism. Stylopods are odd and enigmatic, but they are also one of the most complex and intriguing groups of animals. They are evolutionary marvels that have puzzled and awed generations of entomologists and naturalists, and more surprises should be revealed from future research. It seems quite fitting then for the august Royal Entomological Society to have adopted a stylopod, Stylops kirby, as a representative of the organization. Royal Entomological Society Badge. Copyright Wikipedia Creative Commons. Antlion. The antlions are a group of about 2,000 species of insect in the Neuropteran family Myrmeleontidae. They are known for the predatory habits of their larvae, which mostly dig pits to trap passing ants or other prey. In North America, the larvae are sometimes referred to as doodlebugs because of the marks they leave in the sand. The adult insects are less well known due to their relatively short lifespans in comparison with the larvae. Adults, sometimes known as antlion lacewings, mostly fly at dusk or just after dark and may be mistakenly identified as dragonflies or damselflies. Antlions have a worldwide. In North America, the larvae are sometimes referred to as doodlebugs because of the marks they leave in the sand. The adult insects are less well known due to their relatively short lifespans in comparison with the larvae. Adults, sometimes known as antlion lacewings, mostly fly at dusk or just after dark and may be mistakenly identified as dragonflies or damselflies. Antlions have a worldwide distribution. The greatest diversity occurs in the tropics, but a few species are found in cold temperate locations, one such being the European Urolian nostras. They most commonly occur in dry and sandy habitats where the larvae can easily excavate their pits, but some larvae hide under debris or ambush their prey among leaf litter. Antlions are poorly represented in the fossil record. Myrmeleontiformia is generally accepted to be a monophyletic group, and within the Myrmeleontoidea, the antlions' closest living relatives are thought to be the oldflies, Ascalophidae. A 2019 study finds Myrmeleontidae to be monophyletic, aside from Stilbopteryginae and Palperinae, which form separate clades closer to Ascalophidae. 1. The predatory actions of the larvae have attracted attention throughout history, and antlions have been mentioned in literature since classical times. Etymology. Doodlebug, antlion, doodles and pit traps in the Grand Canyon. The exact meaning of the name, antlion, is uncertain. It has been thought to refer to ants forming a large percentage of the prey of the insect, the suffix, lion, merely suggesting, destroyer, or, hunter. 2. In any case, the term seems to go back to classical antiquity. 3. The antlion larva is often called a doodlebug in North America because of the odd winding, spiraling trails it leaves in the sand while relocating, which look as if someone has been doodling. 4. The scientific name of the type genus Myrmeleo, and thus, the family as a whole, is derived from ancient Greek myrmex, ant, plus leon, lion, in a loan translation of the names common across Europe. In most European and Middle Eastern languages, at least the larvae are known under the local term corresponding to antlion. 3. Adult of Myrmecaloris trigamus. Larva of Myrmeleon amiculatus. Antlions can be fairly small to very large neuropterans, with wingspans ranging from 2 to 15 centimeters, 0.8 to 5.9 in. The African genus Palpares contains some of the largest examples. Acanthoclissus occitanica is the largest European species, with an 11 centimeters, 4.3 in wingspan, and most North American species approach this size. 4. The adult has 2 pairs of long, narrow, multibind, translucent wings and a long, slender abdomen. Although they somewhat resemble dragonflies or damselflies, they belong to a different infraclass of winged insects. Antlion adults are easily distinguished from damselflies by their prominent, apically clubbed antlions. Temporal range: Early Cretaceous, recent. Dystolion tetragrammicus. Dystolion tetragrammicus larva. Scientific classification domain: Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta order, Neuroptera superfamily, Myrmeleontoidea family, Myrmeleontidae. Description: Antennae which are about as long as the head and thorax combined. 5. Also, the pattern of wing venation differs, and compared to damselflies, the adults are very feeble flyers and are normally found fluttering about at night in search of a mate. Adult antlions are typically nocturnal, and rarely seen by day. 2. Males of most species have a unique structure, a bristle-bearing knob known as a pilula axillaris, at the base of the rear wing. The abdomen in males is usually longer than in females and often has an extra lobe. The tip of the abdomen of females shows greater variation than that of males, depending perhaps on oviposition sites, and usually bears tufts of bristles for digging and a finger-like extension. The tip of the abdomen of females shows greater variation than that of males, depending perhaps on oviposition sites, and usually bears tufts of bristles for digging and a finger-like extension. 6. The bottom side of larval mandible with a visible maxilla for injecting venom. The antlion larva has a robust fusiform body, a very plump abdomen, and a thorax bearing 3 pairs of walking legs. The prothorax forms a slender mobile neck for the large, square, flattened head, which bears an enormous pair of sickle-like jaws with several sharp, hollow projections. The jaws are formed by the maxillae and mandibles. The mandibles each contain a deep groove over which the maxilla fits neatly, forming an enclosed canal for injecting venom to immobilize the victim. 7, 8, and enzymes to digest its soft parts. The larva is clad in forward-pointing bristles which help it to anchor itself and exert greater traction, enabling it to subdue prey considerably larger than itself. 9. Antlion larvae are unusual among insects in lacking an anus. All the metabolic waste generated during the larval stage is stored, some is used to spin the silk for the cocoon and the rest is eventually voided as meconium at the end of its pupal stage. 10. Latreille, 1802. Subfamilies. See text. Distribution. There are about 2,000 species of antlion found in most parts of the world, with the greatest diversity being in warmer areas. The best known species are those in which the larvae dig pits to trap their prey, but not all species do this. Antlions live in a range of usually dry habitats including open woodland floors, scrub-clad dunes, hedge bases, river banks, road verges, under raised buildings and in vacant lots. 11. Life cycle. The life cycle of Dystolion tetragrammicus. Apart from pit trap-forming taxa, the biology of members of the family Myrmeleontidae, to which the antlions belong, has been little studied. The life cycle begins with oviposition, egg laying, in a suitable location. The female antlion repeatedly taps the prospective laying site with the tip of her abdomen and then inserts her ovipositor into the substrate and lays an egg. 12. Depending on the species and where it lives, the larva either conceals itself under leaves, debris or pieces of wood, hides in a crack or digs a funnel-shaped pit in loose material. 7. As ambush predators, catching prey is risky because food arrives unpredictably and, for those species that make traps, maintaining one is costly. The larvae therefore have low metabolic rates and can survive for long periods without food. 13. They can take several years to complete their life cycle. They mature faster with plentiful food, but can survive for many months without feeding. 14. 15. In cooler climates they dig their way deeper and remain inactive during the winter. 10. An antlion cocoon on the side of a house. When the larva attains its maximum size, it pupates and undergoes metamorphosis. 15. It makes a globular cocoon of sand or other local substrate stuck together with fine silk spun from a slender spinneret at the rear end of the body. The cocoon may be buried several centimeters deep in sand. After completing its transformation into an adult insect over the course of about 1 month, it emerges from the case, leaving the pupal integument behind, and works its way to the surface. After about 20 minutes, the adult's wings are fully opened and it flies off in search of a mate. The adult is considerably larger than the larva as antlions exhibit the greatest disparity in size between larva and adult of any type of holometabolous insect. This is by virtue of the fact that the exoskeleton of the adult is extremely thin and flimsy, with an exceptionally low density. 16. The adult typically lives for about 25 days, but some insects survive for as long as 45 days. 15. Ecology. Sandpit trap of an antlion. Antlion larvae eat small arthropods, mainly ants, while the adults of some species eat pollen and nectar, and others are predators of small arthropods. 17. In certain species of Myrmeleontidae, such as Dendroleon pantherines, the larva, although resembling that of Myrmeleon structurally, makes no pitfall trap, but hides in detritus in a hole in a tree and seizes passing prey. 18. In Japan, Gatsara jezoensis larvae lurk on the surface of rocks for several years while awaiting prey. During this time they often become coated with lichen, and have been recorded at densities of up to 344 per square meter. 19. The larva is a voracious predator. Within a few minutes of seizing its prey with its jaws and injecting it with venom and enzymes, it begins to suck out the digestion products. 7. 9. The larva is extremely sensitive to ground vibrations. The low-frequency sounds made by an insect crawling across the ground. The larva locates the source of the vibrations by the differences in timing of the arrival of waves detected by receptors, tufts of hairs on the sides of the two hindmost thoracic segments. 20. Pit-building species. Antlion larva extracted from sandpit. Funnel-shaped pits are built by members of just 3 antlion tribes, Myrmeleontini, Myrmecalerini, and Nessoleontini. 21. In these trap-building species, an average-sized larva digs a pit about 2 and 5 centimeters deep and 3 and 7.5 centimeters wide at the edge. This behavior has also been observed in the Vermilionidae Diptera, whose larvae dig the same sort of pit to feed on ants. Having marked out the chosen site with a circular groove, 22. The antlion larva begins to crawl backward, using its abdomen as a plow to dig up the soil. Using one of its front legs, it places heaps of loosened particles upon its head which it then flicks clear of the scene of operations. Continuing in this way, it gradually works its way from the circumference toward the center. 2. As it slowly moves round and round, the pit gradually gets deeper and deeper, until the slope angle reaches the critical angle of repose, that is, the steepest angle the sand can maintain, where it is on the verge of collapse from slight disturbance and the pit is solely lined by fine grains. 23. By digging in a spiral when constructing its pit, the antlion minimizes the time needed to complete the pit. 23. When the pit is completed, the larva settles down at the bottom, buried in the soil with only the jaws projecting above the surface, often in a wide open position on either side of the very tip of the cone. 24. The steep sloped trap that guides prey into the larva's mouth while avoiding crater avalanches is one of the simplest and most efficient traps in the animal kingdom. 25. The fine grain lining ensures that the avalanches which carry prey are as large as possible. 23. Since the sides of the pit consist of loose sand at its angle of repose. 26. They afford an insecure foothold to any small insects that inadvertently venture over the edge, such as ants. Slipping to the bottom, the prey is immediately seized by the lurking antlion. If it attempts to scramble up the treacherous walls of the pit, it is speedily checked in its efforts and brought down by showers of loose sand which are thrown at it from below by the larva. 2. By throwing up loose sand from the bottom of the pit, the larva also undermines the sides of the pit, causing them to collapse and bring the prey with them. Thus, it does not matter whether the larva actually strikes the prey with the sand showers. 24. Antlion larvae are capable of capturing and killing a variety of insects and other arthropods, and can even subdue small spiders. The projections in the jaws of the larva are hollow and through this, the larva sucks the fluids out of its victim. After the contents are consumed, the dry carcass is flicked out of the pit. The larva readies the pit once again by throwing out collapsed material from the center, steepening the pit walls to the angle of repose. 24. Sandpit trap with remains of an ant. Antlion larvae require loose soil, not necessarily, but often, sand. Antlions can also handle larger granular material which is filtered out of the soil during pit construction. 23. The larvae prefer dry places protected from the rain. When it first hatches, the tiny larva specializes in very small insects, but as it grows larger, it constructs larger pits, and thus catches larger prey, sometimes much larger than itself. 24. Other arthropods may make use of the antlion larva's ability to trap prey. The larva of the Australian horsefly, Scaptia muscula, lives in antlion, for example Myrmeleon pictifrons, pit traps and feeds on the prey caught. 4. And the female chalcid wasp, Lasia chalcidia igaliensis, purposefully allows itself to be trapped so that it can parasitize the antlion larva by ovipositing between its head and thorax. 27. Recent research has found that antlion larvae often play dead for a variable amount of time, from a few minutes up to an hour, when disturbed to hide from predators. The method is effective. It increased survival rates in patches that use it by 20%. 28. Furthermore, they appear to have maximized its usefulness. Further increasing the duration is not likely to convey substantial survival benefits to the larva. 29. The closest living relatives of antlions within the Myrmeleontoidea are the oldflies, Ascalophidae. The Nymphidae are more distantly related. The extinct Ararapinuridae and Babinskiidae are considered likely to be stem groups in the Myrmeleontiformia clade. 30. The phylogeny of the Neuroptera has been explored using mitochondrial DNA sequences, and while issues remain for the group as a whole, the Hemerobiaformia, being paraphyletic, the Myrmeleontiformia is generally agreed to be monophyletic, giving the following cladogram. 31. The subfamilies are shown below. A few genera, mostly fossil, are of uncertain or basal position. The fossil record of antlions is very small by Neuropteran standards. However, some Mesozoic fossils attest to the antlions' origin more than 150 million years ago. These were at one time separated as the Palaeoleontidae, but are now usually recognized as early antlions. 17. The suprageneric classification within the Myrmeleontidae is disputed by different authors. Several different schematics have been proposed. Evolution. Taxonomy. Antlion larva trails, doodles, in sand. Thorax and head. With club-shaped antenna, of antlion adult. Larva. 20x close-up of larva. Video of antlion larva trying to catch prey with sand traps and eating a small spider. Video of a larva trapping an ant by throwing sand at it. Myrmecalion, from the Hortus Sanitatus of Jacob Maidenbach, 1491. In popular folklore in the southern United States, people recite a poem or chant to make the antlion come out of its hole. 35. Similar practices have been recorded from Africa, the Caribbean, China and Australia. 36. The Myrmecalion was a mythical ant-lion hybrid written about in the 2nd century AD Physiologus, where animal descriptions were paired with Christian morals. The ant-lion as described was said to starve to death because of its dual nature. The lion nature of the father could only eat meat, but in culture and folklore, ant half from the mother could only eat grain chaff, thus the offspring could not eat either and would starve. 37. It was paired with the biblical verse Matthew 5:37. 38. The fictional ant-lion of Physiologus is probably derived from a misreading of Job 4:11. 37. The French naturalist Jean-Henri Fabre wrote that the antlion makes a slanting funnel in the sand. Its victim, the ant, slides down the slant and is then stoned, from the bottom of the funnel, by the hunter, who turns his neck into a catapult. 39-40. Antlions appear as antagonists in the 1991 life simulation video game, Cement, and, in a giant form, in the Final Fantasy series, Grounded, Terraria, Don't Starve Together, Monster Rancher 2, Mother 3 and in the Half-Life 2 video game series is an unrelated alien insect species sharing sand-burrowing traits with the real antlion larva. The Trapinch, Vibrava, and Flygon Pokemon evolution line is based on an antlion. The fictional Sarlacc from the Star Wars franchise is often compared to the real-life antlion. 41-42. It also appears as a predator in the film Enemy Mine. In the third book of Tove Janssen's Moomins series, Finn Family Moomintroll, a rather large and fanciful antlion appears in the second chapter, depicted as a sand-dwelling predator with the literal head of a lion. A. The verse runs, the old lion perisheth for lack of prey, and the stout lion's whelps are scattered abroad. Author. Gall-inducing insect. A gall-inducing insect is any insect that can cause the growth of galls within plants. 1. There are several groups of insects that meet this description. They include the gall wasps, scales, gall midges, aphids, psyllids and certain species of leaf miner flies. Parthenogenetic generation female gall wasps Neuroteris albipes, from galls on Quercus rover. Galls are growth deformities induced in certain plants by various insects which are mostly specific. Galls induced by insects can be viewed as an extended phenotype of the inducing insect, and gall-inducing insects specialize on their host plants, often to a greater extent than insects that feed on the same plant without creating galls. 2. The gall's form or type depends on what organism is attacking the plant and 2. The galls Parthenogenetic generation female gall wasps Neuroteris albipes, from galls on Quercus rover. Galls are growth deformities induced in certain plants by various insects which are mostly specific. Galls induced by insects can be viewed as an extended phenotype of the inducing insect, and gall-inducing insects specialize on their host plants, often to a greater extent than insects that feed on the same plant without creating galls. 2. The gall's form or type depends on what organism is attacking the plant and where the plant is being attacked. Based on the form, there are 2 classification systems used to identify the cause of galls: causative agents located outside plant tissues, and those agents located inside plant tissues. Causative outside agents include: Krebs gall is caused by surface agents. Fills gall is caused by agents among surface hairs. Fold roll gall is caused by agents within turned over leaf blades. Pouch gall is caused by agents within a cup-like structure that occurs when opposite ends of the infected structure arch upward and form a spherical oval. Causative inside agents include. Covering gall is caused by agents embedded within a gall when plant tissues rise up and surround the parasite. Lysenchyme gall is caused by agents that sink into the plant when the plant cells dissolve away and close around the parasite. Mark gall is caused by agents burrowing within plant tissue before gall develops. This symbiotic relationship is rather one-sided, with not much research verifying any benefit given to the plant species. Therefore, such insects are most likely parasites of their host plant. The insect that causes the gall formation gets an entire microenvironment or microhabitat provided to it, safe from climate and predation, to grow within, and with a rich supply of food formed within the gall for the insect to feed upon. Ecologically, gall-inducing insects, because of their creation of a microhabitat, are often attacked by parasitoid or inquiline insects that take advantage of the excess resources the gall inducer causes the plant to create, often leading to more biodiversity in the ecosystem than what would exist without the presence of gall inducers. Thus, gall-inducing insects are examples of ecosystem engineers. Insect and mite galls. Galls are abnormal plant growths caused by insects, mites, nematodes, fungi, bacteria and viruses. Galls can be caused by feeding or egg-laying of insects and mites. Insect galls rarely affect plant health and their numbers vary from season to season. Control is generally not suggested. Galls usually form during the accelerated growth period of new leaves, shoots and flowers in late spring. Insects or mites damage plants by chewing on them, and their salivary secretions, spit, cause plants to increase production of normal plant growth hormones. Higher hormone production results in increased cell size or cell numbers. These abnormal cell growths are called galls. Mature plant tissues are usually not affected by gall-inducing organisms. The gall keeps growing as the gall-making insect feeds and grows inside the gall. If galls start to form, they continue to form even after the insects die. Most galls remain on plants for more than one season since they become noticeable only after they are fully formed. Formed on leaf blades or petioles. Most common galls appear as leaf curls, blisters, nipples or araniums, hairy, felt-like growths on the upper or lower leaf surface. Deformed growth on stems and twigs. Range from slight swelling to large knot-like growth. Quick facts. How galls are formed. Types of galls: leaf galls, stem and twig galls. Jumping oak gall caused by cynipid gall wasps. Deformed size and shape of buds and flowers. Bud or flower galls. Galls are growing plant parts and require nutrients just like other plant parts. Galls can steal vital nutrients from the plant and affect plant growth. Can be a problem when galls are numerous on very young plants. Damage may occur if there are many galls on branches or present for several years in a row. In most cases, galls are not numerous enough to harm the plant. Most galls do not cause any severe damage to plant health. Control options usually are not needed. Gall numbers vary from year to year. Chemical applications often are not effective because the precise timing of sprays is critical. To be effective, apply pesticide before gall formation begins, but when insects and mites are active. Once galls start to form, it is too late for treatment, as the galls protect the insects or mites. For insects or mites that spend the winter on the host plant, apply horticultural oil before insect activity begins in the spring. Damage caused by galls. How to protect your plants from galls. Caution: mention of a pesticide or use of a pesticide label is for educational purposes only. Always follow the pesticide label directions attached to the pesticide container you are using. Be sure that the area you wish to treat is listed on the label of the pesticide you intend to use. Remember, the label is the law. Insects and mites that form galls. Cynipid gall wasps. Areophede mites. These tiny wasps are the most common gall-producing insects, with various species affecting leaves, stems and twigs. Usually occur on oak and rose. Some examples are jumping oak gall, oak apple gall, oak bullet gall and stem galls on rose. Gall wasps. Stem gall on rose, oak bullet gall, oak apple gall. Spindle gall. These tiny mites live through the winter on their host plant. They begin feeding and form galls in spring as leaf or flower buds open. Mites begin feeding on male ash trees in the spring before the flower buds fully expand. Galls are initially green but turn brown and more obvious in August to September. Can remain on the tree for up to 2 years. The tiny mites enter the male flower structure in mid to late April before the flower seams open. Control is difficult since gall formation begins much before people start to think about pest management. Spraying is effective when done 7 to 10 days before the flower bud is expected to open. Maple bladder gall. Spindle galls on maple and linden. Aranium or velvet galls on maple, viburnum, birch and linden bud gall on birch. Areophede mites, in combination with a powdery mildew fungus, cause hackberry witchesbroom gall. Ash flower gall. Other common galls caused by areophede mites. Ash flower gall, fall. Plant lice or psyllids. Adelgids. Ash flower gall, summer, hackberry witchesbroom gall. Maple bladder gall, aranium gall on viburnum. Bud gall on birch. Hackberry blister gall. Hackberry nipple gall. Psyllids cause some of the most common types of galls in Minnesota. Hackberry nipple gall. Hackberry blister gall. The small insects spend the winter on hackberry or in other sheltered locations such as inside homes. In spring, when leaves appear, they begin feeding and produce galls. Larger nipple galls appear on the lower leaf surface. Smaller, more numerous blister galls appear on the upper leaf surface. These insects are small enough to pass through most window screens and become a nuisance in fall, when they are looking for shelter. Psyllids or jumping plant lice. Early stage, coulis spruce gall. These aphid-like insects are most common on conifers. The most common galls formed by adelgids in Minnesota are the eastern spruce gall and coulis spruce gall. Although alike in many ways, there are important differences between the two galls and their causes. Occurs mainly on Norway and white spruce. This adelgid completes its entire life cycle on a single host. 1/2 inch long green, pineapple-shaped galls are formed at the base of the new shoots. Such galls can weaken stems, with higher chances of breakage under heavy snow loads or high winds. Eastern spruce gall. Aphids and midges. Authors, Jeffrey Hahn and Marcus Erno. Coulis spruce gall. Tree health is affected only if galls are numerous each year. Occurs primarily on Colorado blue spruce and white spruce in Minnesota. This adelgid usually requires 2 hosts to complete its life cycle. Spruce and Douglas fir. Galls are 2 to 4 inches in length, cone-shaped and are initially green-purple, changing to brown as they dry in midsummer. Coulis spruce galls occur at the tips of the new growth. Damage is very rare and only when young trees have numerous galls repeatedly. After forming the gall on spruce, winged adelgids fly to Douglas fir in mid-late summer and lay eggs. Resulting adelgids feed and can produce yellow, twisted needles on the Douglas fir. A winged generation of adelgids is then produced on Douglas fir. They fly back to spruce in the fall, lay eggs and produce adelgids that live through the winter. The females from this population lay eggs in spring to give adelgids that produce gall. No galls are produced on Douglas fir. Coulis spruce gall. Willow pine cone gall caused by midges. Many species of aphids occur on plants and some can produce galls. The most common galls are the stem and petiole galls on poplar, poplar petiole gall, or cottonwood. These tiny mosquito-like insects are responsible for causing irregularly shaped structures on leaves and buds of a variety of plant species. Some of the common midge-produced galls are willow pine cone gall, gouty vein gall on maple and grape filbert gall. Aphids. Gall midges. University of Minnesota Extension. Galls are abnormal plant growths caused by insects, mites, nematodes, fungi, bacteria and viruses. Galls can be caused by feeding or egg laying of insects and mites. Insect galls rarely affect plant health and their numbers vary from season to season. Control is generally not suggested. Galls usually form during the accelerated growth period of new leaves, shoots and flowers in late spring. Insects or mites damage plants by chewing on them and their salivary secretions, spit cause plants to increase production of normal plant growth hormones. Higher hormone production results in increased cell size or cell numbers. These abnormal cell growths are called galls. Mature plant tissues are usually not affected by gall-inducing organisms. The gall keeps growing as the gall-making insect feeds and grows inside the gall. If galls start to form, they continue to form even after the insects die. Most galls remain on plants for more than one season since they become noticeable only after they are fully formed. Formed on leaf blades or petioles. Most common galls appear as leaf curls, blisters, nipples or eriniums, hairy, felt-like growths on the upper or lower leaf surface. Deformed growth on stems and twigs. Range from slight swelling to large knot-like growth. Quick facts. How galls are formed. Types of galls: leaf galls, stem and twig galls. Jumping oak gall caused by cynipid gallwasps. Deformed size and shape of buds and flowers. Bud or flower galls. Galls are growing plant parts and require nutrients just like other plant parts. Galls can steal vital nutrients from the plant and affect plant growth. Can be a problem when galls are numerous on very young plants. Damage may occur if there are many galls on branches or present for several years in a row. In most cases, galls are not numerous enough to harm the plant. Most galls do not cause any severe damage to plant health. Control options usually are not needed. Gall numbers vary from year to year. Chemical applications often are not effective because the precise timing of sprays is critical. To be effective, apply pesticide before gall formation begins, but when insects and mites are active. Once galls start to form, it is too late for treatment, as the galls protect the insects or mites. For insects or mites that spend the winter on the host plant, apply horticultural oil before insect activity begins in the spring. Damage caused by galls. How to protect your plants from galls. Caution: mention of a pesticide or use of a pesticide label is for educational purposes only. Always follow the pesticide label directions attached to the pesticide container you are using. Be sure that the area you wish to treat is listed on the label of the pesticide you intend to use. Remember, the label is the law. Insects and mites that form galls. Cynipid gall wasps. Areophede mites. These tiny wasps are the most common gall-producing insects, with various species affecting leaves, stems and twigs. Usually occur on oak and rose. Some examples are jumping oak gall, oak apple gall, oak bullet gall and stem galls on rose.